Hello, good evening, everybody. In this series of the lectures on genetics, I intend to take about 10 to 12 lectures for the first MBBS students in India. Now, today is the first lecture. It is an introduction to the genetics. Now, these lectures are totally based on the curriculum which of uh, 2019 as per the MCI new curriculum. Okay, so we will just limit to the lectures which are for the first MBBS student. Now I go to the next slide just to show you about the new curriculum. Now the students, they should note this uh, curriculum which is about the revised MCI, okay, curriculums in genetics for 2019, okay. Now this contain many lectures, okay, many lectures, and some are very important lectures to be understood by the first MBBS. And since I am from the Department of Anatomy, I will deal only the structure which is related to genetics. That means I will deal with the structures of the chromosome. Okay, I will not go at the level of the molecular structure for the genes which will be taught by transcriptions or replication, which will be our protein synthesis. That part will be taught by the biochemistry people. Hence, I will limit myself what is the curriculum of the anatomy. Okay, so uh, students should note this. And now I pass to the new slide. I will say the next slide where I will tell you, okay, from where I am, my lectures will be based. Okay, just see this. This is a book, Medical Genetics, okay, and this contain not only the that portion of the genetics which is for first MBBS, but it is almost a complete medical genetics. Of course, elementary, okay, very basic, okay, very basic, which is needed by a medical student. So this book is written by myself and published by the AITBS publisher India. Okay, so all my lectures will be based on this book. Okay, so I acknowledge with thanks to the AITBS publishers. Yes, so we come to an introduction to the genetics. The first question is, what is genetics? Okay, and since you must have observed that the brothers and sisters of the same parents, okay, they resemble to each other. Okay, so the question is, why siblings resemble each others and to the parents? Okay, that means the brother and sisters, they only do not resembles with each other. That means brother is look similar to the sister, or two brothers, they look similar, much similar to each other. But they also look similar, somewhat similar to the parents, sometime more to the father, and other time more to the mothers. Okay, or some characters they show from the mother. Okay, in appearance, I am talking of physical characters. In appearance, they resemble to any one of the parents. Okay, more to one parents and less to other parents. That case may be anything. Okay, now this is because the characters, okay, physical, mental, or biochemical characters are transmitted from the parents to the next generation. That is the reason why they resemble to siblings, they resemble to each other and to the parents. Okay, so the process of transmission of characters from one generation to next is called as inheritance or heredity. So this is the definition, okay, of the inheritance or heredity. That is, it is the process of transmission of characters from one generation to next, and that is the reason why the offsprings, they resemble to the parents and to each other. Okay, now question then comes, what determines the characters? Okay, if your hairs are black, if your nose is sharp, okay, then these characters are determined by on what basis these characters are determined. That is how these characters were transmitted from your parents to you, or these characters are similar in your I mean to say brothers and sisters. Okay, so naturally the question will come in mind that what determines the characters. Moving to the next slide. Now, we know today that these characters are determined by the genes, okay, physical characters, height, color of the skin, color of the hair, okay, the voice, okay. Now, and also not only the physical but the mental, okay, the capability of a person, intelligence level, that are also determined by the genes, and then they are transmitted, okay, to the next generation, that is inheritance. Now, genes are fundamental units of inheritance. You already know from your lower classes, you have read in detail that the genes are responsible for the inheritance. They are the fundamental unit of inheritance and are transmitted to offspring through sperm and ovum naturally. Okay, how it has come to uh, uh, I mean to say in the offspring, they have come because of the union of the ovum from the mother and sperm from the father. Okay, and this process of fertilization has taken place by the fusion of the ovum and the sperm. Okay, so these have come from both the parents. Okay, so the characters have come from both the parents. Whatsoever genes were there on in this present in this ovum and sperms, they will determine ultimately the characters of the offspring. Now genetic is okay. After this background, at least we can make out that genetics is the study of genes. Number one, it is the study of genes. And what are genes? Which determines the characters and which are transmitted from our parents to us. Okay. And second, the genetics is the study not only of the genes but also of the statistical laws, mathematical rules that governs the passage of genes from one generation to next. Okay, from one generation, that means from parents to the offspring. Okay, so what are those statistical law which governs this passage of gene? That is also included in genetics. It is not only the study of the gene. That should be in, I say, you should be clear in mind with this. Okay, uh, you are sitting with your notebook. Just keep on taking the notes. Okay, whatsoever I am teaching you, so that you will keep a concentration and will understand it better way. Okay, this is my suggestion. Okay, just a suggestion only. Now we come to the branches. In the introduction of genetics, we come to now to the branches of genetics. Okay, you must be knowing many of these branches already, must have studied in your schools. Okay, first branch which is of importance is cytogenetics. And what is cyto means cell. Okay, cytogenetics means study of chromosomes which are present within the nucleus of a cell. Okay, so this is the study of chromosome. When you take up, that is called as cytogenetics. Okay, then comes in another branch which is called as molecular genetics. And in molecular means the structure of a gene at molecular level. Because what is the structure of gene, as you know by this time, is that it is made up of the double helix of the DNA molecules. So it is at the level of the molecule. So you cannot see this molecules, a structure of DNA by any means. Okay, not by even electron microscope. You have to understand and deduce or determine it with the help of some indirect method called as X-ray crystallography. Okay, so this molecular genetics is the branch of the genetics which deals with the structure of genes at molecular level. So that is the structure of DNA or RNA. Okay, and that I have told you in the beginning will be taught to you by the biochemistry department. This is not the part of the anatomy. Of course, structure is the part of the anatomy, but the not this part, that is which is at the molecular level, which will be better dealt with by the biochemist. Okay, the biochemist. Now the third branch, okay, of course the cytogenetics is the, I mean say it comes under the anatomy. So we will teach you about the chromosomes, okay, about the chromosomes. Now the biochemical genetics is the metabolic process, study of the various metabolic process which are determined by the genes, okay, by the genes. Say for example, in biochemistry classes, you will learn various diseases which are of the biochemical nature, which are also called as the inborn error of metabolism. Okay, say for example, alkaptonuria, galactosemia, okay, phenylketonuria, so many diseases are there, okay, which are due to the deficiency of one of the enzyme or abnormality of the gene, which leads to the formation of the abnormal enzymes. Okay, so gene is defective, okay, which are responsible for biochemical reactions, right? So this is the third branch, the biochemical genetics. The fourth branch of the genetic is cancer genetics. Okay, lot of research is going on in this branch, cancer. We are trying to determine how the cancer is produced. Okay, as we know somewhat that it is the mutation of the checkpoints gene. There are so many genes in our uh, body which are called as the checkpoint, and they check that the cell division should be normal, it should not be abnormal, and abnormal cell division is the basis of the cancer. Okay, so checkpoints are there in our cell cycle which determines the mm, normal cell division. Okay. Mm. So any interference in the structure of these checkpoints gene will lead to the cancer. The fifth is the developmental genetics. Okay, this will be the developmental genetics. Okay, so there are these branches, many of these branches which are there in this. And lastly, last branch was the, okay, preventive genetics, okay, which is an important, and I will talk a lot about the preventive genetics when I will tell you the last lecture in the series of genetic, that is called as the genetic counseling. Okay, in the genetic counseling. Now this is a next slide where you we will try to understand the importance of genetics in medicine. Okay, importance of genetics in medicine. In recent past, more and more diseases have been recognized as genetic in nature. They are now determined as genetic or recognized as genetic in nature due to increased knowledge of genetics. Okay, in the last 30-40 years, the knowledge of genetics has come, okay, many folds. We A lot of research have taken place in uh, last 30-40 years, and we know a lot now about the genetics. Okay, genetics, and this is because of the knowledge, increased knowledge of genetics, and also because of the reduction of infectious disease. Since when I was a child, the infectious disease were very common because of the unhygienic condition. Okay, secondly, because of absence of the broad spectrum antibiotics, and the medical field was involved in treating these infections first. Okay, so our all mind was diverted towards the treatment of the infectious disease. Now, since the broad spectrum antibiotics are available in country, and we have controlled a lot of the degenerative, I mean say infectious disease. So naturally, with the increase or explosion in the knowledge of genetics, now our concentration is more on the genetic disease, and that's why, okay, many more diseases are now, okay, recognized as the genetic disease. We know now the basis is in the genes, okay, which we were not knowing previously. Now, for example, about 50% of the abortion in first trimester, okay, are due to abnormalities in chromosome. We have come to know that the 50% of abortions which takes place in first 3 months of pregnancies, they are due to the chromosomal abnormality. They are chromosomals are so abnormal that the survival of the fetus is not possible. So it is aborted. Okay, it is aborted. Now 3% of the newborn, those who are born, okay, out of this 100%, the 3% are seen with the congenital malformation, which are genetic in origin. Say for example, cleft lip, cleft palate, okay, defect in fingers, okay, these are the, I mean to say, genetical defect which you see by naked eye in many peoples, and then there are genetical congenital defect or congenital malformation which are inside the organs of the body, okay, which we cannot see, okay, but these 3% peoples are born with this congenital malformation. Now the 2% of newborns are with single gene disorder, okay? They are suffering from single gene disorder or chromosomal abnormality, at least 2% of the newborn peoples, okay, newborn. Now, the 50% childhood blindness. So, you must have learned or read that lot of the children are born blind, okay, or they become blind in the very early stage, okay, an early stage of the childhood. Many of them are mentally retarded, okay. Many of them are born deaf, or and these all disease in 50% cases is because of the genetic disease. In 50 remaining 50%, there may be many other regions, okay. One of the most important may be infection again, but then this 50%, and this is a large number, okay, 50% abnormality in a children is a large number. Now, the most of the cancers are genetically determined. What we have learned in the last slide was that most of the cancers are genetically determined. Okay, and I told you the reason: checkpoints genes. Okay, defect in the checkpoint genes and many other. You okay? In this book, lot of a big chapter is given on to the genetics of cancer, though it is not in the first MBBS course. Okay, it is not. Right. Okay, so now I move to the next slide after learning the importance of genetics in medicine, and that is the reason why the genetics is now in the medical college. Okay, it is in the first year we learn, and you will keep on learning the genetics, okay, throughout your life. That means when you are practicing, okay, then you will have to keep learning this genetics, okay. Now we come on the classification of genetic disease. Okay. Now there are many classes of genetic disease. In this slide, I will talk about single gene disorder, and when we will go to the next slide, okay, then we will learn about the multiple gene disorders or polygenic gene disorder. Then we will also learn about another class which is the chromosomal abnormalities, okay, about the which are due to the abnormalities in the chromosomes, right? So let us come to the first class, that is single gene disorders, okay, where the defect or the disease is due to the defect in a single gene. How many genes are there on our chromosome? They are almost up to 30,000 genes on our 46 chromosomes sitting, okay, 30,000 genes are there, and out of which, okay, even if one is defective, this is going to cause some abnormalities. Sometime it is severe, sometime it is unnoticed, okay. So what is single gene disorder? Single gene disorder, as you will learn in the next few classes, they are classified as autosomal dominant, autosomal recessive, and X-linked. disease. X-linked means sex-linked. X-linked means sex-linked. Because sex chromosome in female and male is X also, except, and there is another Y also, but since most of the genes are sitting onto the X chromosome, okay, so it is called as X-linked uh, genetic disease. So this is due to the defect in single gene, and I will talk to you about this autosomal dominant, autosomal recessive and X-linked individual. There will be a separate lecture for individual of this kind of single gene disorder. Don't just at this stage, you just note down what are the different single gene disorders. Okay, that is sufficient for today's lecture. Now, example of this single gene disorder, which you may be knowing many, for autosomal dominant, there is a disease called as achondroplasia, where the height is short. Neurofibromatosis, where there is tiny pea-like, I mean, say tumors are seen on the skin, okay, and hypercholesteremia. Hyper, that means cholesterol level in your blood increases, okay, it becomes hypercholesteremia. Cholesteremia, this is just a tongue twister for me, okay. Then the second class of the single gene disorder are autosomal recessive, okay. Here, just this is the autosomal recessive, and in this it is albinism. Albinism means white people. You must have seen some people are white, absolutely white, no pigment in their body. So they have to wear the black glasses, okay, because their retina is non-pigmented, okay. Then cystic fibrosis. This is a kind of disease in the lung, okay. Then sickle cell anemia. You must have heard about sickle cell, okay, where the RBC shapes changes, okay, and then they are destroyed. Okay, so this leads to anemia. That means the oxygen carrying capacity of the blood reduces tremendously. Okay, they are always air hungry. I mean, say hungry for the oxygen. Now, X-linked dominant, where the gene is sitting on the X chromosome. In this autosomal means where except the sex chromosome X and Y, the remaining 44 chromosomes are 22 pair, homologous pair. They are autosomes, and remaining part I will deal in great detail about what is autosome and what is X chromosome or sex chromosome. X, this disease is vitamin D resistant rickets, okay, abnormality in the bone formation, which is not due to the vitamin D deficiency. And, and hypophosphatemia, that means deficiency of phosphorus, okay, phosphates. Hmm. Now the X-linked recessive inheritance, and what is dominant and what is recessive, I am going to tell you. If you are not knowing at present, I am sure you must be knowing it. But just don't get impatience. I am going to tell you in great detail about this. Okay, this recessive are the color blindness. You are not able to differentiate between blue, black, or any color. Okay, everything looks black and white. And hemophilia, again the clotting disorder. The blood doesn't clot. So very quickly I will pass to the next slide, okay, because this will be taught to you again in great detail. Now, classification of the genetic disease. In the second classification, it is due to the many genes. There was the first one was because of the defect in a single gene, but here the disease is due to the defect in multiple genes, or at least 2 3 genes, okay, are involved. That's why it is called as polygenic, many genes, okay. And sometimes not only the genes which are responsible for the genetic disease, but along with the genes defect, the environmental factors like the climatic condition or like the diet of a person, okay, they also affect, okay, the inheritance, okay, the disease is because of that. And that kind of the disease we will learn in a separate lecture, what is called as multifactorial disorders, okay. Most of the disease of the adulthood, say for example diabetes, blood pressure, height, color, okay, uh, These cleft lip, cleft palate, they are determined by the polygenic or multifactorial disorder. The third kind of the genetic disease are due to the abnormalities in the chromosome that you must have learned, I am sure. Hmm. This chromosomal abnormality may be due to change in number. There are 46 chromosomes. Even if one chromosome is less or one chromosome is more, this is going to affect the health, okay? And there will be genetic disease, okay? Hmm. Or it may so happen that the part of the chromosome, a single chromosome may break, okay, that will lead to the structural abnormality, okay, or there may be any other abnormalities in chromosomes, say for example ring chromosome formation, okay. So this kind of the structural abnormality within the chromosome, even if their number is normal, this is going to lead to the disease, genetic disease, which is the cause of the genetic disease. And lastly, the acquired somatic genetic disease are another class of disease, and this is due to the chance in the mutation or chromosomal abnormality during the somatic cell division. See, our body is made up of billions of the cell, and they are constantly dividing, and during each cell division, since chromosome has to move to the opposite pole, okay, for formation of the daughter cells. So during the mutation, I say there may take place chromosomal anomalies, or the when the cells are dividing, they duplicate their genes, okay, DNA contents, and at that time the genes may become defective in a person, already born person, and his body, which is containing of billions of somatic cells, may uh, uh, even if one cell is defective, then successively all other daughter cells of that line will be defective. But then in a person, if the mutation takes place, okay, in somatic cell, you understand, it is the cell, okay, except that of the gametes, okay. All cell in our body are somatic cells, okay, except the ovum and sperm. So they are constantly dividing, okay. Right, now I move to the next slide, and this is the last slide, which will tell you what are the recent advances in medical genetics. This will stimulate you to learn, okay, genetics. Don't leave the genetics, is very important subject for your practice, okay. Now, the diagnosis of genetic disease is one of the most important aspect to uh, control or to prevent the genetic disorder in a family. Okay. Now, the diagnosis now available. The technology has advanced tremendously, and we are in position to now know or diagnose a genetic disease before even birth, okay, for birth of a fetus. So there are the say techniques which have come. It is FIS, okay. FIS means fluorescent in situ hybridization, and PCR, you must have heard the name of PCR. This is the time of COVID infection 19, okay? And this is PCR is playing an important role in molecular identification of the virus, okay? The COVID virus is 100%, I mean say exactly identified because of the PCR, because then the uh, nucleotide base pairing is matching, okay, perfectly. If with that of the with the virus, then we say that person is suffering from the COVID-19, right? Now DNA fingerprinting, okay, you must have known it is used for identification of an individual, and in case of disputed paternity, when one is not sure who is the father of this child, then this fingerprinting is done. I will tell you about fingerprinting also, okay, in the last lectures of the series. Then genetic engineering, okay, you can introduce a gene into a virus, I'm not into virus, into a bacteria, okay, or into the animals, so that the product of that gene can be collected. Say for example, if you want to produce the insulin, you inject this gene of insulin into the bacteria which will keep on producing the insulin. Okay. Production of therapeutic drugs, similarly, as I said, insulin, somatostatin, okay, growth hormone, the blood clotting factors, okay, for hemophilia, and the growth hormones, they are those. If a person is not having pituitary or abnormal pituitary or low functional pituitary, okay, the height will remain very short, okay. And if you give the injection before puberty to this person, the height will grow. So these hormones are substitutes which can be given from outside, and thus they are also producing, I mean say genetic knowledge is used in the production of these therapeutic drugs. Okay. Then the genetic treatments, the gene therapy, you introduce the gene in an individual, not in human being right now, okay, but at experimental stage in animals. And you must have also heard about the stem cell therapy. That means those cells which are highly potent, okay, they can differentiate in many kinds of the tissues. They are called as stem cells, okay. And if you inject the stem cell, okay, this may take up the characteristic of the cells where they are introduced and form the new cell, okay, still in the experimental condition. Now the prevention of genetic disease is very important. You have to prevent. If you cannot treat a disease, it is better to prevent it. That should not occur. That disease should not occur, right? That branch is called as prevention of genetic disorder. Okay, and how you can prevent? Okay, by prenatal diagnosis of genetic disease. What is prenatal diagnosis? That means you have to diagnose that disease when still the fetus in the womb of the mother, in the uterine cavity. Okay, and then there are various techniques by which you can, okay, diagnose a disease. For example, amniocentesis. That is a procedure in which the amniotic fluid from the uterine cavity is drawn with the syringe, because this fluid contains many cells from the skin of the fetus. Okay, so we can do the karyotyping or chromosomal study, and we come to know whether there is any defect in chromosomes or not. Then chorionic biopsy is done from chorion. Okay, in embryology, if you have read the chorion, that cells are also fetal in origin, and they are also the source of the I mean say karyotyping. Okay, the analysis of chromosome. Okay, you can take the blood sample also from fetus, and the fetoscope in an instrument by which you can know about the fetus. Okay, sonography, yes, it is very important nowadays. When I was a student, there were no sonographic instrument, no sonography. We were forced to take the X-ray for detection of the abnormality in the skeleton. Okay. Nowadays, sonography can detect many things, many things. The progress of the pregnancy and the abnormalities in organs or anywhere is very well detected by the sonography. Okay, and a person, I mean to say, during the pregnancy, you can screen for thalassemia, whether a person is suffering from thalassemia or not. Okay, that can be very well diagnosed. So the genetic, because of the recent advances, okay, and the research in genetics, we are in much better position to prevent the genetic disorder, which were not few years back. Okay, so this branch is developing very fast, and its future is very bright, okay, of the genetic branch. I think this completes the today's lecture, and then we will go to the next lecture where we will learn about the Mendel's law. And I will, just because Mendel's law is not in your course, but still I will tell you some important facts, because that forms the basis of understanding of the genetic subject. Okay, so in a simple terms, I will limit to myself and will tell you about the Mendel's laws of inheritance. Thank you very much.